I'd like to introduce our next speaker. Once I get on the right page, here we go. Dr. Thomas Page, who is Vice President of Engineering and Asset Development with Fujifilm Diosynth Biotechnologies, USA, is going to talk about advancing the monoclonal antibody therapies of tomorrow. Please welcome Dr. Page.
Okay, great. So, howdy from Texas. I want to thank everybody for coming out to our talk today. And I also want to thank the previous panels for basically doing a great job of setting up exactly the issues that we were confronting as we went forward and we're looking to the future and how we were going to design this capability and bring it on board. So without further ado, we'll jump in. So Saturn is the name of our project, and Saturn is not a planet, Saturn is a rocket. So as many of you may know, Fuji Diosynth has a cell line called Apollo, and the Saturn rocket is what took Apollo to the moon, so that's where the project code name comes from. So really, we have this fancy mission statement, and we basically said, let's go forth and create this competitive capability. Let's add a lot of capacity, and let's really make sure that, we can very flexibly meet our clients' needs. Okay, so that explains also why Saturn. So really, we got to start the process of going about doing this. All right. So what did we have? We had a very senior team that was impaneled from across the 3 sites within Fujifilm, within the UK, North Carolina, and Texas. So this senior team was pulled out and put on this program and then empowered, okay? So really it said, assess all aspects of delivering customer programs, challenge everything, all right? Take the best, throw out what doesn't work from our system, and then innovate where you need to, right? But then put very simply, our US president has a really great way with words. He says, you darn engineers, don't just go off and build me a box, all right? You need to consider the business implications of everything that we're doing, okay? So we have all those functional areas were represented that this core team was governed by a select member of our executive leadership team, so right at the top. So if we had questions, decisions could be made rapidly to move forward on this innovation. So now I'm from Texas. We don't always speak in metaphors and stories, but sometimes we do. So the way I think about it is, this is a very famous football coach, and he didn't care about how fast you were and how much, what you could pick up and all that. If you couldn't be on the field on Sunday, right, you were worthless to him. So the best ability is availability. So how do we do that though? So we're going to use modern risk-based tools to focus on these critical business, regulatory, GMP. So what you're kind of seeing is we're looking at the entire envelope of risk as we go through this process. We need to enhance the risk controls where we need to, and then where we've identified risk controls that perversely increase risk in one of those areas, we're going to yank it out, even if it's a cherished activity or it's someone's pet rock or whatever, it's going out. So then what are we going to output? We're going to output a system of systems so that efficiently and reliably produce monoclonal antibody products on time and to specification, so that we're making predictable supply available, right? That's going to deliver these critical therapies. Yep. Oops. And so this is the output of that. And so the reason I'm showing a video is it's a very dense...
Fujifilm Diosynth Biotechnologies is a world-leading CGMP contract development and manufacturing organization. Our network spans globally with 3 centers of excellence in development and manufacturing in the United Kingdom, North Carolina, and Texas. We support our partners in bringing their biologic, vaccine, and advanced therapies into the clinic and ultimately to commercialization. We're proud to offer our Saturn MAB platform, which is designed for reliable and robust cGMP production of monoclonal antibodies from early phase to commercial. The cGMP monoclonal antibody production has been established in the company's College Station, Texas site. This is a 120,000 square foot state-of-the-art cGMP mAb manufacturing facility with a commercial quality design that allows for up to 24,000 liters of cell culture upstream capacity. The inoculum room features automated vial thaw and has a traditional shake flask scheme. The cell expansion room contains 3 parallel lanes. The single-use bags are customized to minimize contamination risk. All processing is fully closed. The upstream production room is a multi-product area designed to accommodate both clinical and commercial production of monoclonal antibodies. The room features 3 2,000-liter GE single-use bioreactors that can support high-density cultures. This spacious room features single-use chromatography using pre-packed columns for early-phase manufacturing. For commercial operation campaigns, the single-use systems may be replaced with larger chromatography systems and may also be scaled out for a second downstream train. The purification room supports functionally closed processes and features 2 single-use chromatography skids designed for use with pre-packed columns and/or membranes. Nanofiltration is also performed in this suite in a fully closed system with the filtrate proceeding to the next room. Closed processing allows for robust segregation between steps. The bulk fill room features a single-use TFF system that supports a wide range of final bulk concentrations and volumes. It makes use of functional closure and fully closed connections. The bulk filling system supports bags and bottles. Fujifilm Diosynth Biotechnologies is proud to offer its Saturn mAb platform to its partners. It was designed for reliability of manufacturing. It's a platform for life.
So when we talk about platform, and I've already set the stage for you at the breadth of what we were looking at. So what makes up the platform are all these different areas. So many of the questions that came in the previous panels are discussing individual pieces of this as well. So everything from robust processing to the supply chain, the fight against customization and standardization, and how do we leave enough flexibility within the process to take in the variability of the products that we need to handle. How do we deal with the process development, tech transfer process? And then down the road, as we continue to run this platform over many, many different products, there's a battle-tested CMC section that comes out that only the product-specific information needs to change, which we view as a great value to the clients as well. So all this adds up to, this is what we call the platform for life, which I like because I like puns. So it doesn't just mean the life of the patients, these great products that we're putting out the door. It's actually talking about the life cycle of the product itself. So we're viewing that all the way from the initial construct all the way through to commercial manufacturing. So, yep, and we are taking orders now and reserving space in the facility. Okay, so what does it look like? We talked about 3 by 2K. We basically picked the range from about 2 to 8 gram per liter to cover. We're going to start out in the sort of 1 per week range. We can scale up from there. And the other thing we've done is on the downstream chromatography, we're basically cycling within a batch to help reduce the consumable costs for our early phase clients to try to deal with that issue that was being discussed previously. And then later on when you go to commercial, you can actually cycle the column across batches. All right, so how does this fit into our current network? So as many of you know, we have an existing offering for monoclonal antibody production in both North Carolina and the UK. Right? And we have very extensive process development and process validation capabilities. So this slots in. We do an assessment around platform fit, okay? And if it's immediately ready to go and it's commensurate, it will go to the technology translation group that we've created in our UK site. So we've actually started up a whole new facility there to do that in Wilton. If it requires some other more tailored solutions, then it would go into our standard process development, activities, and then could possibly come back into the platform as well at a later date. Which brings me to the function of the group in Wilton. It's not about tech transfer, it's about recipe transfer. So the group in Wilton is actually outfitted with exactly the same equipment that we have in the Texas site. They're using the same connectors so that we have a very predictable outcome from coming through that technology translation layer so that we get a very lean, a very simplified and effective transfer of information, okay? So this is the Wilton Center, so this is the flow. So we've invested quite heavily into this facility as well as into the Texas manufacturing facility. So now I'm going to say, how did we go on this trip, right? So I'm going to try to get through this quickly. I expect there to be some gaps and some questions, so we will be here at the booth afterwards to pick up any of the loose threads, right? So we had a early clean sheet. So of course we were advised not to go off and just build a box, but we are engineers, so what was the first thing we did? We looked at the process and drew a facility wrap around it, and we drew something that was very highly segregated. We segregated the heck out of it. And then we were challenged by some individuals with CRB, Mark von Zwolinski and Mark Peltier, who have been pushing forward quite heavily into the closed processing ballroom area. And they basically said, hey, the 1980s are calling. They want their facility back, right? So if you look at the PICS guidance around closure, okay, it basically comes out in really sort of graphic detail here. If you have a closed process, you could put it outside. Right? You can't unhear that. And there it is. This is the guidance that we're living under, and it is pertinent to antibody production. Okay, so what does that let you do? That lets you do a lot of interesting things. It lets you enable the ballroom. It lets you do things around classification so that you're not getting EM hits that we know are not impacting the product, that are holding it up from being released to the clients. And then on the other side, it does require us to do more around closure, more filtration, et cetera. And it also does allow rolling maintenance. So for example, this facility is not going to shut down. We're going to have a rolling maintenance strategy for the facility, which also increases capacity. So we came up with an advanced modified ballroom concept. So for us, after having done the risk assessments, we maintain a V+ and a V- capability. So the V+ area operates as a ballroom. Even though some of those are in other rooms, we allow the operators to move around. So we capture the greatest flexibility while we think optimizing the risk control around this design. So at that point, when we were looking at a green, we said, hey, you know what, this really looks familiar. I think we have one of these already. So the Texas biomanufacturing facility is an extremely robust facility. We can get into some more of the details around what all that means, but it's there. And we're starting out with an initial 3 by 2K, and we can go up to 12 by 2K. So this is kind of the particulars in. So if we look at the actually pressure cascade, room classification, flows, et cetera, and we take at this and we say this looks like a pretty traditional flow facility, but by just some slight modifications, we can operate that area as a ballroom, and then we can operate that area as a post-nanofiltration area. And that gives us the greatest flexibility with a very optimized risk control as well. So if you're going to do this and you're claiming closure, you can't just wave your hands around and say we have a closed process, right? So the FDA mantra over and over again. If it's not written down, it didn't happen. So when we looked at our high-level documents in our validation program, there seemed to be something missing. So At the highest level, we have a validation project plan around process closure, and it has a whole lifecycle approach to it that leverages the group in the UK to do the closed process development and to collect the initial data using scaled down versions of all these connectors, tubing, methodologies, et cetera. Then we do our closed process qualification in the site, and then we're into a continuous verification model. And then the tool we're using is basically a modified FMEA risk tool around closure. All right, so this is the layout of the process. Okay, so another tool that we use to guide us in this is we did some extensive SuperPro modeling that really looked at where the cost is coming into the client, particularly for these early phase activities. This is where you end up with a small, medium, and large downstream capability, and that allows us to fit the most cost-effective downstream process for any of the clients. Okay, so in conclusion, what we're trying to do is make sort of intrinsically something that's very variable because of so many different clients coming into us, make it routine. So also, can we platform this? Can we have raw materials already set aside and chromatography columns, media, all of that stuff? Now, we do make allowances for clients bringing in their own materials, but we feel that there's a great benefit to our clients to come onto the platform as well. And this platform has no strings attached to it at the end. So if you're on our platform and at some point you leave to do your own thing, you take it with you. And it is optimized around the whole product life cycle. So this is the Saturn team. We've had QA and QC at the highest level dialed into all of these activities from the very beginning. And then we've had a variety of experts in the area of process closure and process architects, with special thanks to the folks at CRB for their great help. So thank you. So